Thank you very much, George, for agreeing to give a Build a Cell seminar, and you can get started.
Okay, great. I'm going to be talking about why and how to build cells, and in particular enhanced cells, cells that are enhanced relative to current ones. My potential financial conflict of interest is in the lower right website. Doesn't really fit on slides anymore. And these are some of the institutions that I have to thank for helping us bring synthetic cells or start to bring them into being. Okay, so why and how? We want novel chemistries. We actually want more robust growth. Most of our synthetic cells are less robust, but our goal is more, and I'll show you a quick example of that. We are quite interested in bioethics and safety, biocontainment, virus resistance, and I'll show you a couple examples of that. We want everything to be safe, effective, which is the FDA's concerns, and equitable, which they often are not required. So we have two real a big fork, which isn't as big as it may seem. It is doing everything from scratch in vitro versus renovating living cells. So this is analogous to the legendary ship of Theseus being rebuilt while at sea. So those are the two branches that I'll start with is scratch in vitro. And nothing is really completely from scratch because we have our knowledge. So even if the molecules are from scratch, our knowledge is there. So now this is just a reminder that for almost every technology, almost every project in biotechnology, there are safety, efficacy, and equity and other ethical, legal components. And a lot of this we've done in collaboration with Jantine Lunshoff. Here she is in one of these authors, these 36 papers that I've collected from our website. And here she is in a few more. And I'm not going to go through all these papers, but I wanted up front to say that this is one of our top priorities. So in addition to the priority for, and somewhat related to the priority for bioethics, there's a priority for communication. It's hard to have a true bioethical commitment without listening carefully to the community and spreading the word and getting practical demonstrations in the hands of others and making sure that that's safe as well. So we've collaborated with the Fab Lab, Neil Gershenfeld, for many years, where they have a program called How to Make Almost Anything. These are MIT courses. How to Grow Almost Anything is our version of it. That is Harvard and MIT and 500 global initiatives, kind of like iGEM. As well, and this is some of the minimal setup for how to grow. Now, my involvement in this starts around actually 1973 when I first started working on transfer RNA, and they started publishing in 75 crystal structure of tRNA, a way to make chips into synthetic. Up to that point, chips had just been used for analytic, and then we made oligonucleotides from chips into an operon in this Nature paper, large ribosomal operon at that. And then Tony Forster, which I think is present in the audience today, and I wrote a couple of papers that were supposed to accompany this Nature paper, but got delayed by 2 or 3 years. But nevertheless, they laid out why we were doing this and how we thought it might go, including doing it entirely in vitro initially. So picking up where that left off, Tony and I wrote these synthetic biology projects in vitro. That was the title in genome research. a few years later, Ueda published this very transformational paper, at least for our field, called PURE. That's the acronym, capital letters. And then Jun Lee and others in my lab published a series of papers where we actually started implementing the vision that we had in the 2004 to 2007 papers. And this is kind of one of the last of that series of papers, just to give you a feeling for why we shifted more towards the Theseus ship approach of rebuilding it from active systems. Is we had a reaction chamber where we have feeding surrounded by feeding and dilution of waste, and as concentrated a solution as we could get, we did curves on almost every component, including the genome, including the overall concentration. And as we got at the higher concentration, there'd be a point of diminishing returns, and that point was about 80-fold more dilute than in vivo. And we thought that you could make a list of things that might be missing from our system. So Tony and I estimated 151 genes, maybe 113 kilobases. And then later we added a few more, maybe 180. This was kind of the diagram from one of our papers. But it could be, it's not just that there's a missing gene or a missing protein or RNA. We even made constructs for all of the pure factors in the Ueda's system and all the ribosomal proteins. And we got up to 21% activity of native 70S ribosomes from the ribosomal proteins that we made in vitro. But we felt that probably wasn't some particular protein that was missing, but it was this concentration issue. And that's still just a working hypothesis, but I'd love to talk about it. By the way, hopefully my talk will be short enough that we can have long, lively discussion. So keep that in mind as you're listening. That this reconstitution of the small and large ribosomal subunit from these messenger RNAs in vitro resulted in this activity that I just referred to. Now, if you're going to rebuild the ship, and not only rebuild it you don't want to make an exact copy, but maybe something wildly different. The question is, how wildly different can you be and still have the ship running all the time, having the cell replicating? So we're talking about replicating cells here. There's all kinds of projects to build a cell where the either doesn't replicate at all, or it replicates something that doesn't have a high density of information as you do in DNA and RNA, or even possibly other information molecules, proteins, tRNA, XNA, and so forth. So here's an example of how you can make a radical change in the system via intermediate. And I'll show you another example in a moment where we change the genetic code radically. But here's where we can not only change the genetic code, but we can free up essentially a whole set of codons. So instead of having 64 codons, we could have 128, because we've got this whole orthogonal system, but it requires all these components. So we have the messenger RNA is orthogonal because it has a different ribosome binding site. The small subunit has a ribosomal RNA that binds to that ribosome binding site. Then you have to couple that to the 50S ribosome so you can have an orthogonal tRNA. So you've got all these mRNA, all the ribosomal RNAs, and the tRNA can all be customized to this orthogonal system. So above here is a particular part of the peptidyl transferase system with the ribosome A and P sites, which, thanks to Suga and collaborators, became particularly evident how to engineer it. Is you've got the CCA is like the most conserved sequence probably in the whole planet. It's exactly the same on all tRNAs and all cells, except for these now synthetic ones, which instead have CGA, but that requires changing these two normally guanines in the ribosome A and P site into cytosine. So now they bind to G rather than to C. And so that can be made orthogonal thanks to work that Mike Dewitt's lab did and other labs. But we used the one from Mike that fused the 23S and the 16S by taking these two loops and turning them into a continuous chain. So now, whatever you do to this orthogonal ribosomal tRNA system doesn't necessarily interfere with the workhorse that's keeping the cell running and keeping this incredible density, like almost 30% solute concentration going. Now we did a quick survey of whether this CGA 3' end of tRNAs were going to be compatible with the tRNA synthetases. And this is some preliminary results. Since then, we have extended that we basically have a whole set of tRNAs that seem to be decoded, seem to be amino acylated just fine with existing tRNA synthetases. So it's quite likely that the main barrier to using a CGA 3' end is not the synthetases, but the ribosome itself, which we can easily change with these two. I hesitate to say easily. We can change with these two mutations and possibly more. And we have done that. Now, I mentioned that our goal is to have robust and rapidly growing systems. This was our favorite naturally occurring system, Vibrio nitrogens, which Henry Lee and Nelly Ostrov published on. It's about twice as fast as E. coli. Most of our engineered E. colis are slower than the 28-minute doubling time, while the Vibrio is faster. And we'd like to understand that. And we'd like to understand the process by which we could make engineered E. coli or other cells more robust. Now, in addition to these heterotrophs, Vibrio and Escherichia, we've also found prokaryotic and eukaryotic algae photosynthetic systems that have doubling times as little as 85 minutes. So examples of each at this kind of doubling time, which is really quite fast for photoautotrophs. And that's thanks to Max Schubert and others. Now, speaking of exponential growth, there has been exponential that I think we're all familiar with in both reading and writing nucleic acids. And this goes back to, my early days as a graduate student, and I think change slope. It started getting faster than Moore's law around 2004-ish for both reading and writing DNA, thanks to chips in the case of writing DNA, and in both cases thanks to multiplexing, where you have multiple optionally barcoded sequences in some kind of selection system or some kind of mixed processing. So, this went from my experience with the first plasmid sequence in 1978 to now the first high quality one error in 10 billion for a human telomere-to-telomere diploid genome that's part of our personal genome project. And the price has come down from $3 billion for a poor quality haploid human genome to in the range of $100 to $200 for a high quality clinical grade diploid genome. And all of these technologies are important for building cells and improving them. And these exponential technologies have brought this 20 million-fold improvement in cost and quality in reading biopolymers, but also we're now writing libraries up to 10 to the 17th, and I'll mention that later on. We've made capsids that are 100-fold more targeted. These are not cells, these are virus, but they have cell-like properties. We've done radical genome recoding. So however you got your cell, you might want it to have the natural if it has a natural code, then it will be sensitive to viruses in principle, but we can make it resistant by changing the code. So we might want to change the polymers, might want to change and we want to do this via something we call machine learning plus multiplex libraries. Even if we're building simple cells, and I think there's one of the debates in this talk is whether we want to build simple cells, but no matter how simple they are, they're often our applications are interacting with complex systems. And we can do that. We can have simple cells interacting with complex systems. And one of the issues here, you can't just delete the histocompatibility antigens because you have natural killer cells which have a receptor KAR that checks for the presence of HLA. And then once you have HLA, then you start running into issues of rejection. So not to digress on this more than one slide, but it's relevant. If we're trying to make transplantable cells or cells that work in concert with complex organisms, then we need to think, we have to think in advance exactly what we want. And we have now made an example of this, not with synthetic cells, but with pig organs through developmental biology. So very heavily dependent upon complex systems, which have various enhancements that have been demonstrated in animals that are either naturally occurring or synthetic biology that are cold tolerant down to -55. These are as whole organisms, severe dehydration down to 3% water, resistant to DNA damage, to immune rejection, to pathogens, cancer senescence. The point is that there's lots of potential enhancements that have been partially demonstrated, and we've got some of them that have already made it into patients, and some of them they're still waiting. Now, in a particular set of enhancements are the ones that we can get by changing the genetic code. So we're changing one of the key information polymers, the genome, the DNA, and we can change it so that of the 64 codons, we can subtract 1 or 3 or 7, and once we subtracted it, we've made the whole replication system independent of that codon. And now we can do new things with it without hurting the host. And in particular, we can do biocontainment, we can have non-standard amino acids and virus resistance. And in fact, it's an interesting vignette, just brief aside, when Tony and I were thinking about what we could do with a synthetic genome, we were initially focused on minimal genomes because we didn't know how to synthesize them, but we wanted to ask, well, what are we going to do with that minimal genome? We don't want to just make one just to show that we can, and we thought that we could make recoding, and originally that recoding was focused on non-standard amino acids, thanks to the work of Pete Schultz, with whom we later collaborated, and that guided us a little bit. But then we also thought of biocontainment and virus resistance. And once we got this whole vision of the applications in mind, we said, oh, well, let's try it both ways. Let's try it with the in vitro system, and let's also try it by recoding, the ship of Theseus, getting the fully working genome to change. So just like we can make that orthogonal ribosome system, here we were using the natural central dogma, but changing the genetic code radically. And then that turned out to work better. We had the two methods going in parallel with a slight bias towards the in vitro, and then eventually this in vivo seemed to work better. And we got all three of these: biocontainment, viruses, non-standard amino acids. And the non-standard amino acids are part of GROW and PEARL, which are thanks to Farron Isaacs and Dan Mendel. So these are, GROW already has non-standard amino acids in human pharmaceuticals making their way into clinical trials. So the planning of this thing is quite interesting. Here's the 64 codons. Hopefully you all know the genetic code by heart. But what's the single letter amino acid code, and the numbers are the number of times that codon is used in the genome. And so the least abundant one, the fewest number of codons in the genome at the ends of proteins are these stop codons, these three stop codons. And we focused on the least abundant of the three stop codons initially. And we did that by genome editing rather than by de novo synthesis, although later Jason Chen and our lab aimed at 2 more codons or 6 more codons. All of them, these are amino acid encoding, all of them focusing on leucine, serine, or arginine, or all three, because they're the only 3 amino acids that have 6 codons. Some of them have as few as 1, like tryptophan and methionine, but these have 6 different codons, which gives us more space to move around codons in so-called synonymous space. Now, as it turns out, they really were pretty synonymous with, a few dozen exceptions, almost essentially no exceptions for the first one, the release factor 1 UAG codon, but dozens for the leucine, serine, and arginine. And this is thanks to Nili Ostrov and others. Fast forward to after Jason Chen's lab made 2 more codons, the 2 serine codons, these TCA and TCG. We leveraged those to test whether we could make them both biocontained and virus resistant and with really tough tests. So rather than just testing it with one new virus, we wanted to test with most of the laboratory strains, categories of virus, and then a whole host of them that we got from bacteria-rich environments like pig feces and sewage and so forth. And we found, in particular, we were looking for viruses that had a robust set of transfer RNAs that they could overwhelm our engineered transfer RNAs. Here we're making a serine to leucine swap, which means these freed up codons, TCA and TCG, which the host doesn't care about anymore, you can make them encode leucine. We used a very powerful phage expression cassette where the phage would make leucine, and we swapped out the anticodon of that phage leucine, so now that would recognize these 2 serine codons. So that meant that either of those 2 codons of the 64 total, either of those 2, whenever they occur, and they occur in essentially every protein of every virus, they get a mixture of serine and leucine, which is essentially fatal. And it's hard to evolve around it because it's affecting so many proteins at once. And any mutagenesis occurrence that was mutagenic enough to fix, all of the proteins simultaneously would also probably damage all the proteins simultaneously. Now, we haven't rigorously shown that we're resistant to all viruses and they can't evolve around it, but it looks very promising. If you look at the far right of this plot, we have undetectable levels even when we put in 10 to the 7th or more plaque-forming units per mil. So we think we're there, but we'll keep poking away at it to make sure. This is thanks to Akos, Svena, and Reagan. This was published in bioRxiv and in Nature. And then since then, we've published a paper on using multi-omics to help debug these genomes. As you do, the more codons you try to change. So 1 was super easy, 2 more was relatively easy. Doing 7 at once was super hard, and we're still in the middle of it, but we're getting close to the end. Now, the other thing, in addition to virus resistance and biocontainment, was non-standard amino acids. And here's kind of an updated list showing one of the first ones that made it into a pharmaceutical product, which was this para-acetophenylalanine, which Ambrx used to add polyethylene glycol to precise locations on protein pharmaceuticals that had too short a half-life. So then in say blood, and then there are photoisomerization, there's redox reactions, both classic ones, but also any reducing atmosphere, reducing environment, like the cytoplasm of a lot of cells, will melt and reduce disulfide bonds, but selenocysteine bonds are more resistant to that. So this is one of the first set of products from Grow Biosciences, was these selenocysteine replacing disulfides. We're also making nucleopeptides, where we have the backbone of peptides and the side chains of the A, C, G, and T and U. And then finally, fluorogenic amino acids, like this one here. This is this nitro based fluorophore, and this Erkin and Michael and Helena helped publish this and get it into a company that could produce these, both the nucleopeptides and the fluorogenics. And when we talk about multiplexing is a way of changing a synthetic cell, or making it more synthetic, or making it keep changing the ship at sea. And this has essentially two axes in the various combinations. It's the number of edits per genome, which can get up into the thousands and millions, or number of edits in a population. So when we did MAGE, multiplex assisted genome engineering, we had on the order of 4 billion edits back in 2009, and some of those cells could have up to 12 edits, and some of them would have 0 or 1. But the library would contain 4 billion, and that was, partial day's work for one student. We call this ML squared, for machine learning plus multiplex libraries. This is, here's some, I don't need to run through all these numbers, but you get things like we had 23,000 different ribosome binding sites and promoters, which at 24 genes would be kind of like sampling, not exploring the entire space, but 23,000 to the 24th power. Similarly, we did lots of combinations of promoters and ribosome binding sites to get at the cis regulatory code, and discovered things like that you would have rare codons would be enriched in highly produced, in high protein production if it's at the beginning of the gene, in contrast to the dogma up to that point. We've explored transcription factor combinations to get cell type and morphology. We've got virus resistance that I just described, which requires at least 18,000 edits. We can do tissue tropism of synthetic cells, or viral capsids, which are even simpler than the simplest synthetic cells. But in each case, in each of these cases, we're talking about things that are not just lipids and cell division molecules, but they actually are information molecules, which contain a lot of information per cell and a lot of information in the cell population, those two axes of multiplexing. Here's Harris Wang. While we were in the process of that, from that 2004 paper, we were making operons of ribosomal proteins and building up into large constructs that we homologously recombined to change the E. coli genome. Harris got antsy and said, I think we need to publish a paper on this. This is pretty cool stuff. And he picked a simple system where in 5 days he he got, in 3 days, he got a 5-fold improvement in lycopene synthesis. Could have been any commercially significant product, but he picked lycopene. It's just required 3 foreign genes, transgenes, and 24 genes to mutate up and down using ribosome binding sites or promoter swaps. And so that was published in 2009 in Nature. So it was 5 years after our first demonstration we could get synthetic biology off of chips, but well before we got 2013, where we got our first genome with a new genetic code. Here, I'm going to talk about protein design, because no matter how we're making our synthetic cells, if they involve proteins, it's nice to be able to radically redesign those. And we have a whole new set of tools thanks to this machine learning plus multiplex libraries. Here's one where we made, it's fairly easy to design proteins that are rocks that basically have one shape, but ones that do shape shifting, like capsids that I already mentioned, and allosteric DNA binding proteins like LAC-I. LAC-I typically binds IPDG. These are harder than making rocks. It binds IPDG and not Gentia BIOS, and we can flip that with a few mutations defined by this ML squared type of method. This predates it a bit. We can flip it to Gentia BIOS being dominant, IPDG being undetectable. Same thing for sucralose, starts out undetectable, and it goes swaps. So now IPDG is the undetectable. And here's a crystal structure of the sucralose, which has 3 chlorine atoms in it, binding to the LAC-I, where there used to be IPDG. We've also, as part of the biocontainment, we've swapped in BIPA, which is a non-standard amino acid, into the active site of synthetases, making the synthetases dependent upon us. We had to carve away all the clashes. You can see these gray side chains are too close to the BIPA. This BIPA is this gigantic amino acid. And so we had to carve those down to smaller amino acids to accommodate it. Once you carve them down, then you can't go back without multiple mutations. This is thanks to Mark LaJoy, Dan Mandela, I've already mentioned, Barry Stoddard and his team who did the crystal structure with it. And then we showed biocontainment went up by about 6 orders of magnitude for each gene that was dependent upon the non-standard amino acid, BIPA, this big amino acid, with compensating mutations around it. So about factors. So by the time you got to 3 or 4 genes that had been changed, now it was undetectable at less than 1 per trillion escape rate. Again, we're going to keep, it's hard to prove that this is completely unescapable, but we'll keep poking away at it. These big libraries, the trick behind getting the big libraries, the ML squared multiplex libraries, require some kind of barcoding. So you can tag them at the beginning, mix them together, put them through a whole complicated set of biochemical reactions or selections. And here's a case where, so we have both protein and nucleic acid barcodes. This is protein barcodes where we show 96 of them, actually a blow up of 8 of them, showing there's essentially there's very strong on diagonal binding. We make pairs of peptide codes and binders, typically antibodies, but we have almost nothing off target. Partly of that is the design, and part of that is empirical. We remove anything that's has a significant off target. And these are used both for neurobiology for making connectomes, but manifold bio uses them for testing multiple tissue-specific medicines in a way that you'll see in just a moment. We've done that. That's for protein barcodes. We also do nucleic acid barcodes. Here for capsids that want to go to specific tissues, we can do something that would be completely unaffordable. Normally, we want to do essentially 1 million primate preclinical trials with 1 million different capsids, or in the previous example, different protein therapies. So either protein peptide barcodes for the proteins because you don't want to, because they don't have a nucleic acid. And if you have a capsid, then you have the luxury of a nucleic acid. But both the same concept. We screen through up to 1 million per primate, and as many tissues as you want to look at. You can find that it's present in one tissue and not in another, or it's got high delivery to all the tissues. Anyway, you can get that information simultaneously on one animal, 1 million constructs, and dozens of tissue types. And here's an example where we improved the previous best on brain delivery from AAV9 to BCAP1. This is 100 times better at brain and 10 times detargeting liver, which is where almost all of these go. So I think I mentioned these specifically for synthetic cells because you want to be able to engineer the surface of the cell. You want to be able to engineer every biopolymer within the cell. And it's not just a small number of mutations. It's potentially a very extensive mutagenesis where we still get high viability of the protein function. So, for example, the 28 amino acids on the surface of the AAV capsid that we think are important for binding to various receptors, both specific and nonspecific. And we wanted to mutate those. It was hard to get more than 4 out of 28 when we started with our best models at the time. And then as we got better at things like logistic regression, neural nets, and other machine learning tools, we could move from 4 out of 28 to 26, 27, and 28 out of 28. And not just one example, but whole libraries of proteins that have conserved functionality, but might have new functions binding to having preference for new tissues. And this idea of machine learning plus multiplex libraries, which I think could be very valuable throughout the synthetic cell community, we've already incorporated into 5 startup companies. Dino and Manifold I've already mentioned. Nabla makes antibodies and G protein coupled receptors. Shape Therapeutics engineers human adenosine deaminase for messenger RNAs using guide RNAs. This has nothing to do with CRISPR. This is just a guide RNA for human deaminase. And Jura, I kind of skipped over it in this slide here, but we have a preprint on this website for how we have documented that we can make up to 10 to the 17th designs in DNA. They haven't been used extensively in selections yet, but it shows where we're going to these incredibly large libraries. And lots of time for discussion. Thank you. I'm thanking the Personal Genome Projects because these are heroes that have volunteered not just their time, but their body parts and medical records and so forth. And this is a reunion about a decade ago where 200 of our alumni from the lab showed up without me knowing in advance this was going to happen. Anyway, period. Thank you. Full stop. I'll stop sharing. Questions?
Thank you so much, George. I'm sure there are questions from the audience, and there's already a few questions in chat. Can you see chat?
I can, yes.
If you could read a question out loud before answering,
Sure.
That's helpful for the recording.
So when you talk about ethics, is the question, should we make life from scratch at all, or can we distribute that technology to all? The answer is both, and probably in that order. So far, most of the biotechnologies we make of replicating systems grow more poorly in the laboratory. Would grow poorly in the wild if they were released from the laboratory. The exception, we need to focus on the exceptions though, and two of them that I've worked on are gene drives, which are intended to spread in the wild by beating the normal Mendelian inheritance. And one of the examples from this talk, which is virus-resistant cells that have a new genetic code, and if they're resistant to all viruses, that should give them a selective advantage. We've made them, I insisted on making them biocontained before we made them virus-resistant, so that even if they did escape, they wouldn't have the BitBay around, because that's only available in the it's only chemically made. Second question, actually very similar to the first. It's a subset of the first one that I already answered. It's specifically about our viruses be a safety risk, and answer is yes. And we need to talk about almost every kind of synthetic biology, especially synthetic cells, for think out of the box as to what could go wrong. Think about it some more as to how you could prevent that. Think about it some more as to what could still go wrong. But in every case, if the benefits outweigh the risks, we have a tendency to do that. So the third question is, how many sense codons do you think we should be able to recode if we built the metabolism from scratch? I would say, how many can we decode whether or not we build the metabolism from scratch? Can you imagine building a synthetic cell with something like 40 different amino acids or more? So the orthogonal system that I described, in principle, has 128 codons instead of 64. That could probably mean 40 amino acids. And then the question is, what do you do with those 40 amino acids? And I think the biggest barrier isn't getting the coding space or even the metabolism. I think it's getting the crosstalk is to have your amino acid tRNA synthetases being able to reject 39 of the 40 amino acids to some extent. And that's already a problem with 20. If you look, there's misincorporation at every level. In fact, John Hopfield did a beautiful paper or two on kinetic proofreading. John just got the Nobel Prize this year for something completely different having to do with AI. But I think it's good to remember this kinetic proofreading for synthetases, for polymerases, and so on, ribosomes. The next question is, how close are we to making a synthetic cell from the bottom up? The simple answer is, I don't know. I think the point of this talk is saying we don't have to, I don't think we need to be religious about whether we're making it from the bottom up or the top down. In fact, various hybrids, solutions there. I like focusing on what we want to do with it once we have it. And most of my, out-of-the-box applications doesn't seem to matter how you got there. But I'm definitely open to discussion about that. And the last question in the chat is, now there's two. Ribosome biogenesis in cell-free systems, what are the technical barriers you see? What general tools do we need to invent, so forth? I've singled out one, but maybe it doesn't deserve to be singled out, but I think it's a big one, which is getting the concentration right. We would like to have ribosomes that can build ribosomes, which means you have to have all those other things. The, post-translational modification is not a luxury, it's actually part of the core. We need chaperones, they're not a luxury. This is by empirical experiments. It still is a modest number of peptide bonds that you have to make, on the order of 8,000 peptide bonds per ribosome to break even, maybe twice that to start exponential growth. And this has been challenging. I don't know anybody who's done that yet. And it could have to do with the being off by 80-fold in the concentration. Let's see. You mentioned a number of startups that come out of this work, recommendation for resources, those who like to get started. I don't know of a book or a website offhand that lays this out, but you know, we could have a longer conversation about that. I think don't think that it's trivial. You know, I don't try to pretend that it's trivial. And some of it has to do with where you're located and fads and things like that. So it's not for the faint of heart. Here's a question about bringing a synthetic cell. Should have been reading off everybody's name, but I don't know. It's a toss-up between anonymity and credit, but I think everybody can see the chat. Biggest roadblocks for FDA approval for synthetic cell. I actually think that there's not any gigantic roadblocks. Even fairly radical new technologies, like I think the messenger RNA technology behind the COVID-19 vaccines was pretty new, and it got approved in record time. By record time, I mean from the point of the emergency starting to the vaccine being available. Obviously, there were decades of research that went into getting it ready for that year, but it took about a year to get FDA approval. That involved lipid nanoparticles and messenger RNA modifications and so forth. I don't think that our synthetic cells face a steeper battle than that.
George?
Yeah.
Before you move on to the next chat question,
Sure.
We have an in-person question from Tony.
Oh, great. Okay.
Hi, George. Great to see you. Great talk. Very interested to hear you putting the 4 nuclear bases into the polypeptide. And I know Steve Benner's done a lot of work on the importance of the backbone. And I'm wondering what you see happening with those or expect to happen with them with the peptide backbone.
Right. So we're not necessarily trying to make the tRNAs that we can replicate, although that has been done. There are chemically synthesized tRNAs that have been shown can be copied into conventional nucleic acids with polymerases. We think that at a minimum, these peptide backbone with nucleobase side chains could be used for recognizing nucleic acids in protein DNA or protein RNA interactions that currently are done by less elegant rules than the Watson-Crick rules. So that would be an interesting application. But you know, I think it's like a little baby. You don't really know what they're going to grow up to be. Thanks for the question, Tony.
Thanks, George.
All right. If there are no in-person questions, then there is chat questions still.
Yeah. The last chat question that I see is, in the protein design campaigns you described, does machine learning guide selection of which residues to diversify or just assign weights to a library which is empirically constrained? I mean, the machine learning uses everything that it knows about protein, either protein structure, 3D, or in many cases, protein sequence without the 3D, where you're just looking at multi-sequence alignment. And it can use a lot of other things as well, but those are 2 classic things that we use for diversification. It produces, yes, both which residues to diversify and assigning weights to them. And it works incredibly well. I mean, I just can't overstate how grateful we are about this revolution in protein design that we had been using, Rosetta and similar molecular, energy-based energy minimization protocols. I've been using them since 1973, actually. But this game changer. Last question is, do you think we'll see another minimal cell soon, like JCVI mycoplasma, but based on another organism? Maybe. I think part of it is, I think people are more and more focused on what you're going to do next after you have it. And what typically happens is once you have a minimal cell, you start adding doodads onto it. And that you could consider that a chassis. Chassis is kind of the term that we used in Senberg in early days of synthetic biology and to this day. And that's the reason to minimize it is so you can expand it back out again. In principle, you might want to develop a minimal version of every basic cell type, but if it's too minimal, it no longer has the features that made that particular species exciting. Like the Vibrio nitrogens, grows fast. Acetobacter might be used for production of amino acids, et cetera, et cetera. So I suspect that we will keep pushing on maximal cells at least as much as we work on minimal cells. That's just my bias. In other words, cells that have lots of metabolic potential and you can flip them around easily. Follow up on ribosome biogenesis question. Do you think it would be better to implement it in lysates or pure systems? And you've tried both is part of the question. Yes, we've tried both. I don't have a strong opinion. And I would add, the third one is keeping it inside a cell where it's highly concentrated and fully functional, but that's, I guess, cheating in some certain senses. I like the pure system. We've modified it various ways. We made it so that you can make all the ribosomal proteins and so forth. But it is typically a little less efficient. It's probably a little further away from having ribosomes make ribosomes. But if you really want to declare victory on ribosomes making ribosomes, it probably should be pure.
So if there are no more questions in chat, we have a few more minutes left for in-person questions. My question, and I know that's not something you actively work on, but what's your feel for division? Are we going to get a synthetic cell division based on natural proteins like FTSZ systems, or do we need something entirely new? Because we're getting those contraction rings and they don't finish. If you were to bet, what's your division system?
Well, I think there's going to, just like with minimal cells, it's a similar thing. I think it's going to be pushed to make things more and more minimal. And I think we've seen some of this from Jack Shostak's work and colleagues, where you can kind of get imperfect cell division without any proteins at all. At the other end, we're going to want to have efficient division where you get, equal partitioning of the cell and all its components, including any kind of information molecules, if you will. And I think that's going to require more sophisticated systems. So I think what we're going to be happiest with, both will be interesting, but I think the things that are going to lead to applications are going to be the more complicated protein systems that we haven't even tried yet. So we haven't failed either. Yeah.
Thank you. And one more question in chat.
Let's see. Do you think it's possible to de novo design a smaller, less complicated biomolecule that functions as a ribosome? I love this question. Actually, I love all the questions, but it seems like if anything is a vestige of early days, this is it. But it's not early enough. So it's not early enough so that it has, we might imagine the original ribosome was smaller just because of the information content of the ribosome is so vast, it couldn't just like pop out of the abiotic soup. And so it'd be really nice to have all the intermediates along that, whether or not you're interested in in prebiotic life or origin of life, it would be kind of cool to have all these intermediates from say, oligonucleotide soup, which would indicate that smaller is possible. Certainly there are peptidyltransferase centers that are smaller, but that completely ignores all the importance of having an A, a P, and an E side, and the messenger RNA, all that stuff. My gut feeling is that not only was there something less complicated in the past, there's something less complicated in the future, but I don't know how much smaller it could be and still have the kind of rates that we're interested in. You might even imagine something smaller and faster. That would be really cool, but I haven't seen a good proposal for that yet. So here's a question: has anybody looked at the JCVI mycoplasma ribosome, which has fewer ribosomal proteins? I don't think it's missing very much, and most of the ribosome is RNA anyway. But I don't know if anybody's looked at its structure. Oh, and he says asking everybody. Another last question. As a fourth-year student, a PhD, eager to return to SynBio, what advice on what to focus on while completing PhD to be ready for SynBio careers afterwards? Well, I've described a few skills that I am heavily biased towards. I'm not really biased necessarily just by history. I think we tend to pick tools that we think will be of general usefulness, like reading, next gen and reading and writing and editing. But I also mentioned a few that have to do with large libraries and selection, in vitro systems. And so I think any of those would be useful skills. And then, of course, completely outside of the topic of this course are things having to do with this lecture, is things having to do with developmental biology, which I think would also make you an effective candidate in a particular category of SynBio.
I think that's all the questions in chat, and I was told you have a hard stop right now, so any last closing questions? I just want to say thank you so much for the talk. And thanks everyone for a wonderful discussion, and we will let George go before someone yells at me for keeping him too long.
No, he's going to yell at you, Kate. I won't let that happen. Okay, but thank you all, and look forward to more discussion of this fascinating topic. Take care.
Thank you. Thanks everyone, and have a great Monday.
Yeah.
Bye.